Welcome back in another video tutorial from microbiology with Shagni. So we have discussed about the Agrobacterium tumefaciens mediated gene transfer in an introductory manner in our previous video. So in that video we are going to discuss about the topic in a little bit detail. Okay, although it is not the last video regarding the Agrobacterium tumefaciens mediated gene transfer. Now this Agrobacterium mediated gene transfer method is mainly famous among creation of transgenic plants. Okay, so whenever scientists want to create transgenic plant, they need this bacterium, that is Agrobacterium tumefaciens. Although some scientists also use Agrobacterium rhizogenes that we have discussed in our previous video, but that is not too much famous as compared to Agrobacterium tumefaciens. Okay, so as we all know, for any kind of gene transformation we need vectors, right? So here there is a natural vector which is a plasmid, we all know, of 200 kb length called as Ti plasmid is used, and this Ti plasmid is present in the Agrobacterium tumefaciens, and that is why Agrobacterium tumefaciens is too much popular during any kind of transgenic plant formation. So today's video, our goal will be to discuss about the Ti plasmid, okay, so, and how we can genetically engineer the Ti plasmid so that we can use this Ti plasmid as a vector for introducing our gene of interest that is comparatively good for plant instead of the tDNA that Ti plasmid used to naturally transfer inside the plant and facilitates the formation of crown gall disease due to the overproduction of auxin and cytokinin. So today we are going to discuss this. So let's come to the first slide. So the Ti plasmid. So first thing is, it is a vector that is routinely used in transgenic plant, okay, and it is derived from a soil bacterium called Agrobacterium tumefaciens. This bacterium causes a disease we all know, crown gall disease, in which the infected plant produces uncontrolled growth called tumor or gall. So these galls are normally developed in the bases of the stem of the plant, and these bases are known as crown, that is why the crown gall disease. Okay, so the key of the tumor production is the plasmid that is present inside the Agrobacterium tumefaciens, that is the tumor inducing plasmid, or in short Ti plasmid. When the bacterium infect a plant cell, a part of the Ti plasmid, that is tDNA, that is the harmful for plant, transferred and inserted apparently more or less randomly with the plant genome. The region of the Ti plasmid that insert into the host plant is called tDNA or transfer DNA. The gene whose product catalyzes the tDNA transfer reside in a region of the Ti plasmid that separate from the tDNA region itself. Then this gene is our virgins we have discussed in our previous video. So the tDNA region, after inserting inside the plant cell and incorporating or integrating with the plant genome, used to produce three hormones. One is auxin, another one is cytokinin, another one is opine. The first two one, auxin and cytokinin, involved in overproduction of phytohormone, right? Because they are usually a phytohormone, they facilitate the overgrowth of plant, and we all know that anything that is overgrowing leads to the formation of tumor. So this auxin and cytokinin hormone produced by tDNA are the main culprit of the crown gall disease. Okay, and the third thing that are produced by tDNA inside the plant cell is the opine. Okay, this opine is a unusual amino acid whose whose synthesizing gene is present in the tDNA region. That is why TI plasmid infected plant is used to produce opine unusual amino acid, but plant cannot utilize it. Okay, so as a result, but instead there is a opine catabolism region present in the TI plasmid. So it is believed that the opine synthesized by the infected plant cell is going back to the bacteria and supports the TI plasmid's growth, because their opine catabolism region is present. So TI plasmid can utilize the opine as its nutrition. So the TI plasmid, upon transferring the tDNA inside the plant nucleus, facilitates the tumor production as well as its own growth. So this is a kind of very useful relationship for TI plasmid along with the plant. So that is the normal thing that is going on. And remember, here the main and key factors are played by virulence region. Okay, they are producing various kind of weird genes. These weird genes not only involved in cutting the tDNA from the two borders, but also a type of weird gene that we are going to discuss in our next video, used to transfer the portion of the tDNA inside the nucleus of the plant cell. Okay, so weird gene also play a important role, and as we have discussed that this left border and right border are nothing but flanking directed repeat of 25 base pair sequence, and they play a important role in transfer of the tDNA. Okay, so this is the important components of TI plasmid. University often asked to draw a TI plasmid and mention its important structure. So you have to simply draw this picture: virulence region, origin of replication, opine catabolism, right border, opine, cytokinin, auxin, left border. Okay, so that is the normal thing that is going on. Now our plan is to simply removing this So our goal here is to simply remove these bad genes out and insert our gene of interest here. Okay, so but unfortunately it is not so that much easy as we thought, because there are no such kind of unique restriction site because there are no such kind of normal restriction site present here, so that we can cut out the tDNA region. Okay, and there are various important portion such as the opine that is itself important for the TI plasmid. Okay, so we cannot completely cut out the tDNA and insert our gene of interest to facilitate the transgenic plant formation. So what can we do then? So as a substitution of that problem, or as a solution of that problem, scientists developed a procedure that is commonly known as co-integrative plasmid formation. So what is this? Let's discuss. The first step that they have performed is they simply digest the TI plasmid with the help of restriction endonuclease, that is available for the TI plasmid and convert the TI plasmid into a disarm plasmid which contains only the L gene. And here remember that this L gene nothing but the V regions. Okay, so they have convert the TI plasmid into disarmed TI plasmid. Now they have generated a intermediate vector artificially that can be produced with the help of PBr322, or you can also take the help of plasmid to generate your intermediate vector. And remember, this intermediate vector should be smaller than the TI plasmid. Okay, and in that intermediate vector there will be a inactivated virulence gene, okay, L minus. There will be a insert of interest, kanamycin resistant gene, spectromycin resistant gene, opaline synthesizing gene, and the R, that is opine utilizing gene. So they have created this intermediate vector inside E. coli, okay, by simple cloning procedure that we know. Okay, now inside the E. coli they have performed a recombination. Let me take a color first. Okay, now inside the E. coli they have performed a recombination between the disarmed TI plasmid and intermediate vector, and it is obviously a site specific recombination between the virulence gene. So site specific recombination occurs and it leads to the formation of co-integrate plasmid. Okay, so that one, co-integrate plasmid. Now this co-integrate plasmid is simply extracted from the E. coli. Okay, now with the help of normal transformation procedures such as electroporation, micro injection, or gene gun, what we have, we will simply transfer this co-integrative plasmid in Agrobacterium tumefaciens. Okay, now once we have transferred our co-integrative plasmid inside Agrobacterium tumefaciens, we will infect a cut segment of plant tissue. We will infect a cut segment of plant tissue, okay, with the help of the bacteria containing our co-integrative plasmid. Okay, so after the generation of that co-integrative plasmid and transformation of this co-integrative plasmid inside the Agrobacterium tumefaciens, we will simply infect a cut segment of the plant tissue with the help of this co-integrative plasmid containing bacteria. Okay, now if we place this cut segment of plant in a medium containing kanamycin, then those plant cell will undergo cell division that have successfully acquired the co-integrative plasmid because it contains, let me zoom it, the kanamycin resistant gene, right? So in that medium, all the plant cell that have developed or grown into callus, okay, or also known as clumps, are all co-integrative plasmid containing plant cell. Okay, now this callus or clump, or commonly known as calli, can be easily infected into plant into soil, and this calli will lead to the formation of transgenic plant. That is how a normal transgenic plant develops. Okay, so I will again repeat the entire transgenic plant formation once again. So here it is the TI plasmid, and the problem here is TI plasmid is too large to manipulate, and we cannot also cut them into small TI plasmid because This plasmid contain various important sites that is important for their replication, that is important for their transferring, and that is important for their infection. Okay, so we cannot completely digest TI plasmid, and also there is not too much restriction site present in TI plasmid. Only such some unique restriction enzyme site are present in TI plasmid. So digesting TI plasmid is very difficult, and as I have told you that just in removing the entire tDNA and inserting the gene of interest here will not solve our problem, because in the tDNA there is a opine. So if we do this, the TI plasmid cannot perform its own metabolism, because this opine will integrate inside the plant nucleus and produce the opine that will in turn go back inside the Agrobacterium tumefaciens, and this opine will be utilized by TI plasmid with the help of the R gene, that is opine utilizing gene. So if you our plan of just cutting out tDNA will not work. So as a way out, we have developed the co-integrative plasmid concept. So where first of all we are digesting the TI plasmid with the help of restriction endonuclease, and leads to the creation of disarmed TI plasmid, in which we have only the virulence gene. Now we will develop our intermediate vector inside E. coli. Okay, and inside the E. coli we will perform a recombination. Okay, this recombination will leads to the formation of co-integrative plasmid here. And also another important information, that here the virulence gene in case of intermediate vector will be inactivated, because try to think that if a here disarmed TI plasmid already contain virulence gene, and now if via recombination we will generate co-integrate plasmid, and intermediate vector also contain a active virulence gene. So as a result, in the co-integrate plasmid we will have two activate virulence gene, and as a result it can facilitate the overexpression of our gene of interest that can be dangerous for the plant. That is why we will take a inactivated virulence gene, but just for homology, because we all know that recombination, site specific recombination occurs between two homologous sequence. So we need that homologous sequence. That is why we have incorporate L minus in the intermediate vector, and that will produce our co-integrate plasmid. Now this co-integrate plasmid will be transferred inside the Agrobacterium tumefaciens with the help of standard transformation procedures such as electroporation, microinjection, according to our choice. Now after the creation of Agrobacterium tumefaciens containing our plasmid of interest, okay, we will simply infect our Agrobacterium containing plasmid of interest in a cut segment of plant tissue. Okay, so after we have infected cut segment of plant tissue, if we keep that cut segment of plant tissue in a medium containing kanamycin, only those plant cells will undergo cell division that have successfully acquired kanamycin resistant gene. That is why they are capable of dividing. So this plant cell will grow and leads to the formation of callus. That is a part of botany. We just simply need to remember the term callus or clump, and this callus or clump will in turn have the ability to induce shoot formation and root formation. Okay, so if we introduce this calli in soil, this soil leads to the formation of the transgenic plant. That is how the entire thing going on. Okay, so let's move back to the slide. So here I have discussed about the TI plasmid manipulation procedure that I have discussed with you with the help of the diagram. And here the last line is important, that typically a single copy of tDNA region inserts into a plant genome where it segregates via a normal Mendelian fashion at meiosis. That means 1 will be segregated into 2, 2 into segregated into 4, in that way. So that's it for today. Today we have discussed about the TI plasmid, how TI plasmid usually causes infection in a overall manner. We are going to discuss about that particular topic, that is the infection mechanism, in a deep in our upcoming videos. And we have discussed about the TI plasmid manipulation, how we can generate TI plasmid in such a way so that we can use TI plasmid for our purpose. They had this point is important, that why we cannot use normal manipulation technique, why we need to form co-integrative plasmid, because TI plasmid are too large to be easily manipulated and cannot be readily made smaller, because they contain few unique restriction site, and because much of the plasmid is necessary for either its replication or for infection or transfer process. That is the reason we need to go for the co-integrative plasmid formation. So I hope that this video will be helpful to you, and if it is, please hit the like button, share with your friends, and subscribe to my channel to get more video like that. So thank you guys. I will again come back with another video from Agrobacterium tumefaciens, and we will discuss about the Agrobacterium tumefaciens for more 3 video, and then we will wrap up this topic. So thank you for listening to this class.